So today we will talk about platelets. So basically, in the blood cells we have WBC, RBC, and platelets. So WBC, RBC are completed. So now we will talk about platelets. Basically, all these three cells are manufactured in red bone marrow. Okay. So red bone marrow is the place where platelets, WBC, and RBC are prepared. So there is a stem cell. The stem cell will prepare one type of cell. Then different types of specialized cells will be prepared from that cell. Okay. So now we will talk about platelets. Basically, the other name of platelets is thrombocytes. The other name is thrombocyte. Likewise, WBC leukocytes, RBC erythrocytes. So these are the names which are uh, practically given in the book. Okay. So whenever the term erythrocytes are given, you can understand that it is related to the RBC. If it is related to the WBC, it is given leukocyte. Basically, two names we have to uh, remember always. So whenever infection in the body, the WBC counts will be raised. Okay, WBC count will be raised. So that condition is known as leukocytosis. Leukocytes mean WBC. Cytosis means increase in the cells of WBC. So leukocytosis means increase in the increase in the count of WBC. So if it is decrease in the count of WBC, what does it mean? It is mean leukopenia. Leukopenia. What is the other name? What is the name for low WBC? Leukopenia. So ल्यूकोसाइटोसिस ल्यूकोपेनिया, ये दो नाम हमें याद रखना चाहिए, क्योंकि ल्यूकोपीनिया कहा जाता है डब्ल्यूबीसी काउंट के कम होने की वजह से, एंड ल्यूकोसाइटोसिस डब्ल्यूबीसी का साउंड अगर बढ़ जाए तो ल्यूकोसाइटोसिस, ये हमें रिपोर्ट्स में देखने को मिलता है. फ्रॉम दिस वी हैव टू अंडरस्टूड. एंड अनदर टर्म वेरी regularly used for blood cancer, that is leukemia. Leukemia means blood cancer. Leukocytosis means increase in the count of WBC. Leukopenia means decrease in the count of WBC. And leukemia means WBC or blood cancer. Okay. Now we will talk about platelets. Basically, platelets doesn't have any that much of work in the body. Okay, that much type of work in the body. But whenever you have bleed, you can able to see the work of platelets. Okay, let's discuss about basic things about platelets. Platelets का दूसरा नाम क्या है? Thrombocytes. What is the other name of platelets? That is thrombocytes. It is not a complete cells, then it is like a pieces of cells. Okay, I told you that it is prepared in the red bone marrow. ये कहाँ बना है, कहाँ prepared होता है? Red bone marrow. So इसका जो बेसिक नेम है उसको कहा जाता है मेगा कैरायोसाइट्स, मेगा कैरायोसाइट्स. ओके. और द shape is oval. They are small, they are oval in the shape, and they are pieces, okay, which are circulating in the blood circulation. The lifespan is 5 to 9 days. Okay, the lifespan is 5 to 9 days. If we not utilize for 5 to 9 days, what will happen? Uh, the liver have a protein that is called thrombopoietin. Okay, it is a hormone which is secreted by the liver to increase the production of platelets. So here platelets have one type of hormone which is present in the liver, which is result if the platelets are less in count and not utilized from 5 to 9 days, then after they will die. Okay, then again we have to prepare the platelets count. So increase the platelet count. So then the liver will produce one hormone, thrombopoietin, which will increase the platelet production. What is the normal value? 1.5 to 3 lakhs. Okay. Okay. Sometimes it is 5 lakhs also. Okay. 1.5 to 5 lakhs also normal range. Basically, one term is used that is thrombocytopenia. Okay. For low platelet count. If the platelet count has decreased, then 1.5 lakhs, 1.5 lakhs. So it means that the platelets count in the body is decreased. So the term used for decrease of platelets, that is thrombocytopenia. Thrombo means WBC, platelets, cyto means cells, penia means decrease. Okay, decrease in the cell count of platelet. Okay. सो यहाँ पे हम प्लेटलेट्स के बारे में डिस्कस कर रहे थे. प्लेटलेट्स का दूसरा नाम क्या है? थ्रोम्बोसाइट्स. Okay, first thing you have to remember like this. Second thing is that it is prepared in the red bone marrow. So basically सब सेल्स स्टेम सेल से ही बनते हैं. स्टेम सेल्स के बाद एक स्पेशलाइज्ड सेल्स बनते हैं. डब्ल्यूबीसी बनाने के लिए एक सेल, आरबीसी बनाने के लिए एक सेल और प्लेटलेट्स बनाने के लिए एक सेल. ठीक है. तो अब प्लेटलेट्स को बनाने के लिए जो स्पेशलाइज्ड सेल है उसका नाम है मेगा कैरायोसाइट्स. उसका नाम क्या है? मेगा कैरायोसाइट्स. ये स्मॉल होते हैं, यानी प्लेटलेट्स जब भी ब्लड स्ट्रीम में आ जाते हैं स्मॉल होते हैं, ओवल शेप, यानी ओवल शेप के होते हैं. और वो प्लेटलेट्स जब आएंगे पीसेस की तरह नजर आएंगे. आपको ऐसा दिखता है कि कुछ टुकड़े बॉडी में घूम रहे हैं. ठीक है. और ये इनकी जो लाइफ स्पैन है 5 to 9 डेज. अच्छा, ये 5 to 9 डेज के अंदर यूटिलाइज नहीं हुए, ये तो डेड हो जाएंगे. डेड होने के बाद फिर कैसा? फिर बॉडी की रिक्वायरमेंट होगी. तो बॉडी की रिक्वायरमेंट को पूरा करने के लिए लीवर के पास एक प्रोटीन है जिसको कहते हैं थ्रोम्बोपोयोटिन. एक हार्मोन है जिसका नाम है थ्रोम्बोपोयोटिन. ये क्या करेगा? आपके सेल काउंट को बढ़ाने के लिए मदद करेगा. ठीक है. ये सेल काउंट को बढ़ाने के लिए मदद करेगा. अब ये नॉर्मल रेंज क्या है प्लेटलेट्स का? 1.5 लाख से 3 लाख या 5 लाख तक नॉर्मल रेंज. ठीक है. 1.5 लाख से 5 लाख तक नॉर्मल वैल्यू है. अगर प्लेटलेट 1.5 लाख से कम हो जाए तो उस टर्म क्या है? थ्रोम्बोसाइटोपिनिया. ठीक है. द टर्म फॉर यूज फॉर प्लेटलेट्स डिक्रीज, दैट इज थ्रोम्बोसाइटोपिनिया. सो लेट्स टॉक अबाउट फंक्शन. वेरी वेरी इंपॉर्टेंट. फंक्शन क्या है? फंक्शन इसका functions. Plates are necessary for hemostasis, which means prevention of blood loss. So basically platelets का काम क्या है? blood loss को बचाना. So what is the function of? So first basically I will explain in English. The same topic will explain in the Hindi also. Okay, for Hindi students. So 2 times I will revise. So then easily you can grab it. Okay. Yes. So for everyone, the platelets counts are same in the kids as well as adults. Because one thing you have to remember that the body is the same working condition. Okay, in the adult also, in the kids also, because the normal functions are same. Okay, baby also breathing, baby also sitting, he also digestion capacity. But thing is, his body is not that much big as adult. Okay, so that is the same. That is the only different thing. But all the functions in the body are same. So that's the reason the normal values of the blood is Same in the baby as well as in the adult as well as in the. So we can't tell that 400 ml, 500 ml, but it is here given that per microliter. Okay, per microliter, whatever you are taking sample in that microliter, we have 1.5 lakh to 3.5 lakh. So you have to count down like that. You have to multiply with 400 ml. Then it's what is the value of 1 microliter? Convert into the liter. Then you will get the result. Then you have to multiply with 400 ml. So that is the value you will get. Okay, I think you understood. So basically, what is the function? Here, main topics. Okay, the things are very important, but also here the understanding of the concept, what is platelet and what are the functions when they decrease, is very important. Okay, so come on the topic. So here the functions, what is to maintain the body balance, to prevent blood loss. One short answer, what is the function of platelets? To prevent blood loss. Okay, so here we have the detailed mechanism of platelets. Let's talk about. One is vascular spark. What is meant by vascular spark? Let's talk about a large vessel such as an artery or vein is severed. The smooth muscle in its wall contracts in response to the damage. Platelets in that area of the rupture release serotonin, which also brings about the vasoconstriction. The diameter of the vessel is thereby made smaller, and the smaller opening may be blocked by a blood clot. If the vessel did not constrict first, the clot that forms would quickly be washed out by the force of blood flow. So here, what happened? Uh, in a case, large arteries are there in the body. You have number of arteries, veins. So if the artery or vein are damaged, okay. So here we have the artery. One artery is there supplying to this muscle. Okay, so it is damaged. Here rupture happened because of someone hit or traumatic injury who fell down. Injury happens. One piece of uh, vein or one blood vessel, wall is ruptured. What will happen? Blood will enter into this area and muscle. Okay, so so here we have the what is the muscle? If the muscle in that area will contract, what will first thing will happen? It contracts because of the response of the damage. Because damage happens, the muscle will contract. Okay, first thing will happen. Then what will here? There are platelets in every part of the body. So what will platelets do? Platelets release serotonin. We'll talk later about serotonin. Okay, so serotonin will release serotonin. What will serotonin do? Serotonin will vasoconstrict. It will constrict the blood vessels. It will constrict the blood vessels to remove the blood very rapidly. Okay. So if they are heavy loss, okay, what will happen? The blood is entering into the muscle. What will happen? First, serotonin release. First step, platelets, what will do? It will release serotonin. Then the blood vessels contract and the blood will not enter into the smooth muscle. First, basic physiology. Then second thing, what will happen? If the vessel is not contracting, okay, okay, what will happen? Then the clot is made by the platelets. Platelets will occupy the space and it will block this area so that the blood from the blood, the whatever blood is present in the blood vessel, so it will not enter into the smooth muscle. So this is the second thing what will happen. If platelets is released serotonin, then it is the vasoconstrict happen. So the whatever blood clots is made by the platelets, okay, so it will release from the blood vessel. Okay, understood. So again, I will repeat it that Here we have one artery. Here we have smooth muscle. Okay. Here we have the artery and here we have the smooth muscle. So basically, if rupture happen, so the muscle will constrict first. Smooth muscle is contracts. What will happen? Smooth muscle will contract. First thing. Second thing, what will happen? The platelets, whatever present in that area, will release serotonin. Serotonin constrict the vessel. Second thing. Third thing, that the platelets will make blood clots to fill that area. Okay. Third thing. Okay. If the blood vessel did not constrict, so the blood clots made by the platelets will attach that area and the blood will not enter into the smooth muscle. It will take time. If the vasoconstriction happened, so the blood clots which are made by the platelets will wash away from the increase, because vasoconstricted increase the blood pressure. So from the blood pressure, the blood clot will vanish away. Okay, understood. So this is the vascular spasm. Uh, in that platelets will first work upon vasoconstriction, not on the first choice is clot. First choice is vasoconstriction. Okay. So next, what is the next? So I will repeat in the Hindi. Then we will continue platelets फ्लक्स. तो यहाँ पे वैस्कुलर स्पाज्म में होता क्या है, जो भी आर्टरी है, अगर मान लीजिए यहाँ पे रप्चर हो गया मार लगने की वजह से, कुछ कोई भी चीज हो गई, तो यहाँ पे मसल्स जो रहते हैं पहले मसल से कॉन्ट्रैक्ट हो जाते हैं, ताकि अगर ये कॉन्ट्रैक्ट ना हो या अगर फैले हुए हैं तो ब्लड आके जमा हो जाएगा. तो मसल्स कॉन्ट्रैक्ट होके ब्लड को रिटर्न भेजने की कोशिश करेंगे. फर्स्ट स्टेप ये हो जाएगा, ऑटोमेटिकली हो जाएगा. ठीक है, जब भी ब्लड वेसल में कोई रप्चर होगा. सेकेंड थिंग क्या होगा, प्लेटलेट्स, यहाँ के जो प्लेटलेट्स होंगे वो एक हार्मोन को या एक सब्सटेंस, केमिकल सब्सटेंस को रिलीज करेंगे, उसका नाम है सेरोटोनिन. सेरोटोनिन का काम क्या है, वैसो कंस्ट्रिक्ट करना, ब्लड वेसल्स को नजदीक करना, उसको सुखाड़ना. ठीक है, ताकि ब्लड वहां से पास हो जाए, ब्लड जमा ना हो. ठीक है, तो सेरोटोनिन क्या करेगा, वैसो कंस्ट्रिक्ट करके ब्लड को वहां से जल्दी उसको दूर करेगा. इसी दौरान प्लेटलेट्स क्या करेंगे, अगर ये तरकीब अगर काम नहीं आए तो हमें दूसरा प्रॉब्लम सॉल्व करने के लिए कुछ दूसरी तरकीब भी होना चाहिए. तो प्लेटलेट्स क्या करेंगे, प्लेटलेट्स का काम ही जहां पर भी ब्लड लॉस होने वाला है वहां पे क्लॉट बनाना. और वो क्लॉट कैसा बनेगा आगे देखेंगे हम लोग. क्लॉट बनाना. तो जब भी प्लेटलेट्स क्या करेंगे, फौरन क्लॉट बनाने की कोशिश करेंगे. इस दौरान अगर वैसे कॉन्स्ट्रिक्ट हो गया, तो वैसे कॉन्स्ट्रिक्ट हो गया तो ब्लड क्लॉट जो है वो वैनिश हो गया, वहां से चले जाएगा, क्योंकि ब्लड अब रुक रहा नहीं. और रुक नहीं रहा तो ब्लड वहां पे फिर यहाँ पे क्लॉट की जरूरत ही नहीं, क्योंकि ऑलरेडी यहाँ पे आपको क्या है, ब्लड वेजल्स स्मूथ मदद से कॉन्ट्रैक्ट है, तो ब्लड यहाँ पे रिलीज हो जा रहा और ब्लड यहाँ पे लॉस भी नहीं हो रहा. तो इस वजह से यहाँ पे क्लॉट की जरूरत नहीं है. तो प्लेटलेट्स का क्लॉट जो बनाया हुआ है वो ब्लड प्रेशर की वजह से वहां से वैनिश हो जाएगा. तो थोड़ी देर के बाद ये हील भी हो जाएगा, कुछ घंटों के बाद, कुछ दिनों के बाद ये हील भी हो जाएगा. ठीक है, तो यहाँ तक हम लोगों ने समझा. नेक्स्ट काम क्या है, फंक्शन क्या है ब्लड प्लेटलेट्स का. सेकंड है प्लेटलेट फ्लक्स. When capillaries rupture, the damage to slide to initiate the formation of blood clot. The rough surface, however, causes platelets to change shape and become sticky. This activated platelets stick to the edges of the break and to each other. The platelets form a mechanical barrier or wall to close off the break in the capillary. Capillary ruptures are quite frequent and platelet flux, although small, at all that is needed to seal them. Would platelet flux be effective for breaks in large vessels? No, they are too small, and though they do form, they are washed away until a clot begins to form that can contain them. Would vascular spasm be effective for capillaries? Again, the answer is no, because capillaries have no smooth muscles and cannot constrict it out. So, यहाँ पर here we have the second thing, platelets plug. When small arteries rupture, so here one function we talked about, vascular spasm will happen. Okay, second is platelets flux. If vasoconstriction not happen, so what will platelets do? Platelets will make one type of seal like this. So like this, they make the seal, rough surface becomes sticky. Okay, the activated platelets, because platelets are inactivated during the circulation. Whenever blood rupture happen, the platelets activated and it will stick together. So assume that this is the one platelet, this is the one platelet, this is the one platelet, which are circulating in the blood vessels. But after the rupturing of the blood vessels, what will happen? The all the platelets will stick together like this, like the chain, like the chain, like the chain, like the chain, like this. So it will, okay, understood. It will protect the blood vessels to go outside from the blood vessels. It will protect the blood to go outside from the blood vessels. So, but this is in the case of small arteries. But if they are large arteries, this is the large artery, like aorta, cerebral artery. These are the large arteries we have in the body. Okay. So what will this this function will not work at all on this condition, on this situation. So what will other type of mechanism, that is chemical clotting, will happen. This is very important. Okay, this is very important. So again I will repeat in the Hindi. Platelets flux, तो यहाँ पे मैंने आपको ऑलरेडी बता दिया था कि जब भी ब्लड वेजल्स में रप्चर होगा तो वहां पर तीन चीजें होती है। एक है स्पाज्म, और एक है प्लेटलेट्स सेरोटोनिन को रिलीज करते हैं, और तीसरी चीज प्लेटलेट्स कुछ वहां पे प्लक्स बनाते हैं, क्लॉट जनरेट करते हैं। क्लॉट कैसा होगा? प्लेटलेट्स एक दूसरे से चिपक जाए। प्लेटलेट्स क्या थे ब्लड वेजल्स में? इनएक्टिव थे। प्लेटलेट्स क्या थे? इनएक्टिव थे। लेकिन अब क्या होंगे? वो एक्टिवेट हो जाएंगे जब भी ब्लड वहां पे रप्चर होगा। तो ब्लड रप्चर हुआ, प्लेटलेट्स एक दूसरे से कनेक्ट हो गए, कनेक्ट होके एक जाली की शक्ल में बना। जाली जिस तरह होती है ना, मछली या फिर कोई भी हम चीज पकड़ते हैं, इस तरह वो जाली बनाती है। लेकिन ऐसा राउंड शक्ल में, इस तरह नहीं, ट्रायंगुलर रेक्टेंगुलर के शेप में नहीं, बल्कि ये शेप में यहाँ पे जाली बनाते हैं ताकि वहां से ब्लड लीक ना हो। लेकिन क्या ये बड़े आर्टरीज, बड़े वेन्स के लिए हेल्पफुल होगा? तो आंसर इज नो। बड़े आर्टरीज, बड़े वेन्स के लिए हेल्पफुल नहीं होगा। वहां पे दूसरा मैकेनिज्म, क्योंकि हम डेली हमारे बॉडी में स्मॉल वेजल्स, ब्लड वेजल्स बहुत सारे रप्चर होते रहते हैं। जैसा कि आप गिर गए, मार लग गया या कट गया, ठीक है, किसी ने धक्का मारा, आप गिर गए, गाड़ी से गिर गए, मार लगा, चोट लगा, तो वहां पे छोटे छोटे ब्लड वेजल्स होते हैं, रप्चर हो जाते हैं। लेकिन बड़ी आर्टरी जैसे रेडियल आर्टरी है, रेडियल वेन है, अल्नार आर्टरी है, हाँ, यहाँ पे ब्रेकियल आर्टरी है, तो ये बड़े बड़े वेजल्स है, ब्लड वेजल्स। देखेंगे एक मरतबा, लेट्स सी द ब्रेकियल आर्टरी एंड let's see one artery, big artery, okay, small arteries. So you can see that this is the brachial artery. This is the brachial artery. Okay, this is the brachial artery, which is largest artery. If it is cutted, what will happen? So is platelets have the capacity to block that artery? In that mechanism, it is not possible. So what is the other mechanism platelets have to block this type of arteries are damaged? अब हमारे बॉडी में है तो छोटे छोटे जो रप्चर्स होते हैं, देख सकते हैं आप, ये है यहाँ पे छोटे छोटे आर्टरीज, एकदम थिन आर्टरीज है। The thick arteries, veins. Okay, this is the cardiac wave image. Okay, you can see all arteries, veins. Okay, so I hope you understood. Next, we will talk about third function of platelets, that is chemical clot. This is the very important. Okay, so the stimulus of clotting is a rough surface within a vessel or a break in the vessel, which also creates rough surface. The more damage there is, the fast clotting begins, usually within 15 to 120 seconds. The clotting mechanism is a series of reactions involving chemicals that normally circulate in the blood and others are released when a vessel is damaged. The chemicals involving clotting platelet factors, chemicals released by the damaged tissue, calcium ions, and the plasma proteins, prothrombin, fibrinogen, factor 8, others synthesized by the liver. This clotting factor also designated by Roman numbers factor 8, which is the factor 8. Vitamin K is necessary for the liver to enzyme synthesize prothrombin and several other clotting factors 7, 9, and 10. Most of our vitamin K is produced by the bacteria that is live in the colon. The vitamin is absorbed as the colon absorbs water and may be stored in the liver. So basically here, if the basic mechanism fail, okay, but it fails, okay, that is the reason we have chemical clotting. So what will happen? This is the blood vessel. This is the big artery or big vein ruptured happen. Here is the ruptured part. Okay, so this is the ruptured part. Understood? So this is the big artery. Okay, so break break the artery, make the surface of the blood vessel rough. So what will happen? First thing, rough. You have to remember that. Second thing is that the clotting will happen in 15 to 120 seconds. So it is depend upon the rupture. If the rupture is more, the clotting will start early. If the rupture is less, so it will take more time. Okay. So what are the chemicals? So first is platelet factors. So we have platelets, they have supportive factors. Okay, if the platelet is there, like I am uh, I have I have something that belongs to me. If it is lost, first I will try to bring or capture to from myself only. So if it is not possible, then I will call my friend, call my brother. Okay, all will gather and we will try to solve that problem. So first, this will happen. So platelet, not only platelet factors, their family will come and try to solve this problem. What are those families? Here we have platelet factors. One is platelet factor. Okay. Next is chemicals released by damage. Here we have the damage tissue. They will also release the chemicals. Okay, chemicals. Then is calcium ions. Calcium is also responsible. Next we have plasma protein, prothrombin, also PT. Okay, next we have fibrinogen, and factor 8. Okay, and vitamin K, very important. Vitamin K is responsible for. Next we have factor 7, 9, and 10. This is very, very important. Basically, we have first is platelet factors, next is damaged tissue chemical, then is calcium, prothrombin, fibrinogen, factor 8, 7, 9, 10, and vitamin K. So these are the all family members will go through this area and want to solve this solve this problem. So let's see how it will solve. Chemical clotting is usually described in three stages. We are listing the figures. So here we have the one stage. Okay, chemical clotting stages are there. Stage one. What will happen? Stage one. Okay. So what will happen? The platelet factors, chemicals that are damage, tissue thromboplastin factors, calcium ions will reach that area. And what will they do? Platelet factors will connect each other. Tissue thromboplastin will connect each other. Other factors are connect each other. So they will make like this clot. Okay. So this is the rupture. Mm, you can see that here. This is the skin. So this is the cut down. Okay, cut down. What will happen? So here we have the blood vessel. This is the blood vessel. Okay, big artery. So cut it down. So what will happen? Blood enter into the outside of the blood vessel. What will happen? First stage. This is the platelets. Okay, platelets will come together. Fibrin. Then here we have the clot. They are come together and make a clot. So pack that area. Okay. Second stage. What will happen in second stage? Second stage after come together, then what will happen? Prothrombin activator from prothrombin activator. This will activate prothrombin, calcium ions will join together. So they will convert prothrombin into thrombin. Okay, they will convert prothrombin into thrombin. Thrombin means clot, bigger clot. Okay, then we have the stage 3. Then what will happen? Thrombin from stage 2 will convert into fibers. Fibers will uh, fibers mesh will covered with that area, and calcium ion and factor 13 fibrin stabilization factor will cover. So basically all the stages will step by step will improve, okay, the clotting factor, and it will basically will cover that area and will heal that area. So this is the basic thing we have to remember. Okay. So this is the function of chemical clotting. So basically, we need to remember that clotting factors that are very important. Okay. So here again, I will repeat. I will summarize. So basically, what will happen whenever big artery or big vein are ruptured? So what will happen? The platelets and relatives of platelets. Likewise, you have to remember. Okay, platelets and relatives of platelets will activate. What are the relatives? I mentioned the name. You have to remember these things. Very, very important. Platelet factors, chemicals released by the damaged tissue, calcium ion, fibrinogen, okay, and prothrombin, factor 8, 7, 9, 10, okay, and vitamin K, very, very important. So basically, these are the things we have to remember. So after stage by stage, the the things will improve and the repairing of uh, surface of the area will be same. Okay. So here is the diagram. Liver produce factors 5, 7, 8, 9, 10, 11, 12. So don't confuse that factor. What is the factor 6, 7, 8, 9? Because basically they are the clotting factors. So they are the family members of platelets. Okay. So here platelet factors, chemical from damaged tissue, all will mix in calcium ions. So first stage is remember these four things. Prothrombin activator. This is next text is prothrombin activator is released from the liver, calcium ions again connected with them, then thrombin will form. Next, again, third stage, thrombin will convert into fibrinogen will come and it will activate, and again factor 8, 13 will come, calcium ions also add up, then fibrin will happen. Okay, so means fibrinogen, prothrombin to fibrin. This is the stage, this is the changes of the clot. Prothrombin is the early stage, fibrin is the last stage. You have to remember. Okay. Next is prevention of abnormal clotting. So then we have the summary. So we will summary and we will start the next lecture. So next lecture is the heart. So basically we'll continue lecture. Please join.